Hello, I'm your host, Ray Dogum, and welcome to VibeCast. VibeCast is Vibe Bio's weekly podcast where we explore some of the hottest topics in drug development, biotech investing, and technology innovation. Join us to learn, imagine, question, and help us identify and develop solutions together. Our guests today are doctors Christopher Reyes and Eyram Adjogatse, and they're on the show today to speak about their company called Bloom Science that's focusing on developing therapeutics by harnessing the gut-brain axis. Welcome, gentlemen. How are you today?
Doing great. Thank you so much for having us.
Awesome. Fantastic. I want to get to the part about what is the gut-brain axis, but first let's start off with introductions. And maybe Christopher, if you want to go first and explain, who you are in the company, what's your role, and maybe sort of what led you to founding Bloom Science.
Yeah, absolutely. So my name is Christopher Reyes, and I'm CEO and co-founder of Bloom Science. Yeah, my background is actually in structural biology. I got my PhD from UCSF in structural biology, biophysics, particular X-ray crystallography, which really was kind of the initial journey into biotech. My initial job was at Biogen Eyram when they're here in San Diego, doing monoclonal antibody drug development. And I just had the great fortune to spin out one of my programs focused on cancer stem cells into a company called Eclipse Therapeutics. And that was acquired within 18 months of initiation, which is fantastic. Stayed with the acquiring company, a public biotech company from Australia called Bionomics. And then I got the itch to do it again. And so that's kind of what led me to Bloom. And I could probably spend the whole podcast talking about just this one journey, part of the journey. But, Bloom was really kind of born out of this, I think, this interest in a couple of different things, right? Number one, how do we just frankly rethink a little bit how we approach neurological diseases, right? It's really challenging, such high unmet need. And frankly, particularly when we started Bloom, 4 or 5 years ago, not a huge amount of research or focus in drug development space. Now we see that. Two, how do we match some really novel biology to that thesis, right? I mean, my last company was cancer stem cells. It's kind of a little small offshoot of oncology. So I was really looking at something different. And that's where this whole focus on the gut-brain axis. And I think what I saw in terms of the ability to bring a few things in, novel biology from gut-brain axis, and then an approach from looking at the microbes in our guts, which have, depending on who you talk to, we've either co-evolved with them or they have co-evolved with us, clearly play a really key role in our foundational biology. And so that was the thesis around Bloom is let's combine these two different things. The third really key part, which I will not take any credit for, I will take credit for finding it, which came out of Elaine Shao's lab, Dr. Elaine Shao at UCLA. And her research was really the foundational research which Bloom was started on. And that's this idea of can we reverse engineer a clinical intervention that we know modulates the gut microbiome, in this case, ketogenic diet. So it's not looking at dysbiosis, and we'll get into all those different things and terms, but I really like that idea because it's very much de-risked the opportunity. So all put together, that's how we started Bloom.
That's fantastic. Thanks for sharing that. Eyram?
And then on my side, I always had this really long-term interest in drug discovery. Originally, I saw myself as the scientist who would invent the cure that would save a lot of people. So I originally trained as a pharmacist, studied at the University of Nottingham in the UK, completed my pharmacy training, and then immediately after that started a PhD in drug discovery. So in a similar way to Chris, I covered structural biology as part of my PhD alongside computational chemistry and other disciplines. But during that process, I really started to understand that it wasn't just the science that determines whether a drug makes it to market or not, which caused me to have a bit of a career shift. After that, I joined a consulting firm, consulting in competitive intelligence and strategy, mainly for some of the largest pharmaceutical companies and some mid-sized companies. But then when I left that company, I wanted to take another angle and focus more on the business development side of things. So I started off by helping universities to out-license their inventions. Later on, I took a shift to focus on the startup space, and that's where I met Chris, and eventually that's how I became Bloom's Director of Business Development.
Very interesting. Yeah, it sounds like you guys both have really robust backgrounds in biotech and drug development, so it sounds like, you sort of were very well prepared to be here at Bloom Science. So let's dive straight into
Yeah.
gut-brain axis, if you don't mind. I think it's really interesting. And like you mentioned, there are so many important things about the gut that we're just learning about
Mhm.
now in a way, and how it affects the CNS, the central nervous system, as well as could impact the symptoms of many different diseases. So
Absolutely.
maybe you can just start on like, what is the gut-brain axis?
Yeah, I mean, and this is really the central thesis for Bloom science, where we are developing therapies to target the biology that exists within this gut-brain axis, really to develop, we think, very novel and exciting treatments for patients. And so when we talk about the gut-brain axis, I mean, we have these various different systems in our bodies. We know, for example, like there's a lot of nerve endings in our gut. This is how we communicate whether we're satiated after eating, and that's a bidirectional signal, right? Like we fill our tummies say, "Hey, I'm full," sends it to the brain turns off hormones and says, "Stop eating," right? Vice versa. You can just, you can actually say like, "I don't want to eat because something's going on," sends it down to the gut, and you can have different like excretion of, digestive enzymes, etc. So that's like the simplest example of how we have these two systems interacting. Well, we take it as the additional component where we add then in what the gut is full of, which is microbes. Now, of course, our body is covered in microbes. Like literally everywhere you can imagine, there's probably microbes. And so, oh my gosh, I don't
Not to
know.
scare anyone that's,
Yes,
afraid
exactly.
of bacteria or something. This is a natural thing, obviously. Yeah.
That's correct.
And I think most of the people listening to this will get that.
Yes, exactly. Yeah, and I think it's a really important point to highlight, which is that these are not necessarily, when we talk about bacteria, we tend to think like, oh, it's scary, infectious. That's the first thing we go to. But again, the microbes in our gut, on our skin, I mean, nasal cavities, everywhere, our oral microbiome, again, we've co-evolved. They process a lot of the environmental signals. In the case of the gut, that's our food intake, and break it down into this kind of metabolic profile that feeds into so many key important pathways. So very specifically, one thing at Bloom that we're very much looking at is how does that metabolic profile, how's the gut microbiome influence that? Because all that metabolic profile feeds into this really key neurochemistry, right? And in the cases where you have, say, an epilepsy, which is one of our programs, you may have overexcited neurons. They're firing too much because they're missing a key inhibitory molecule. So if we can change the metabolic signature, basically coming from the gut, can we force that system in our brain to have more, to produce more inhibitory molecules, which is GABA? Can we actually add that, drive that, signal to the brain? So there's some really interesting opportunities there. And then there's another axis, which is of course inflammation. And when we talk about the gut, it's a really essential part of that gut-brain axis that is kind of emerging, which is the regulation of neuroinflammation, but really starting from these pro-inflammatory signals, which of course comes from interaction with the gut. So if you eat really bad food and you get an infection from something, of course you're going to have this immune response. Similarly, our good bacteria help modulate that immune system and keep it primed and ready to react. And what we're seeing, I think, is low-grade inflammation in a lot of diseases over decades may play a very big role in some of these neurodegenerative diseases, maybe even neuropsychiatric diseases.
Very interesting. And so you're not particularly looking at how you can change the composition of the bacteria in the gut necessarily. You're just trying to understand how these signals work so that you can create a therapy or a drug that can sort of imitate or mimic the effect a certain population of bacteria could do. Is that right?
Yeah, it's a great question. And I think that's frankly, the answer has evolved as we understand more.
Oh,
So
awesome.
the short answer is no, we are actually, we've identified specific species of bacteria that are responsible for a particular metabolic signal. And then we've screened our library of microbial strains to find the best within those species to drive whatever the functionality is. And so that kind of matches the old school biotech. You screen against a functional assay, find the best, and then you move it forward. What we've discovered as we've done all of this, of course, is all these new interactions, right? And so frankly, it's true. We are both, identifying and screening and developing therapeutic strains. We're also engineering some of these. We're also engineering some functionality in. But in reality, the names of these bacteria don't really matter that much. It's their function. And so, yes, so the answer is kind of yes to both. We are delivering specific therapeutic strains, but because they have that specific functionality. Eyram, I don't know if you wanted to add to that.
Yeah, I think the other thing that we can add is so we're a microbiome company, so we have several microbiologists on staff, but we really have several people from a traditional drug discovery background. And whilst it's been a very strong paradigm that's focused on single target or single mechanism of action drugs, by taking this approach, we're actually able to identify bacteria that can hit several mechanisms of action. That's either a single species that does that, or when we develop our therapies, we often form consortia of bacterial strains. And through that, we can hit several different targets or different pathways simultaneously. And I think that's really key when we're looking at some of these complex neurological disorders.
Thanks for sharing that. Yeah. So what would you say is your most exciting drugs or therapies right now in your pipeline today?
Yeah.
Can you kind of describe what's going on there?
So our lead program called BL001 is the first program to come out of that reverse engineering of the ketogenic diet. And we're developing that for Dravet syndrome, which is a rare pediatric developmental epilepsy. We will expand into other developmental epilepsies as well. And then kind of serendipitously, we also are developing it for ALS. We did some early research, seeing some utility and some murine models in ALS. And so we're actually also moving into ALS. And we're really excited because we just completed our phase 1 and we're planning on starting our phase 2s, both in Dravet and ALS in 2024. So that's super exciting. And then we have a really robust discovery, platform that has a number of different programs, both from taking kind of a similar approach to what generated BL001, but then also our whole engineering where we're just like, okay, now we know so much more than, say, 4 years ago. Let's take those specific pathways, and that can be production of bioactive molecules, or it can be the reduction of bioactive molecules that aren't favorable, right? It can be a combination of all of those. And we're engineering that into bacteria as therapeutics. And so that's also quite exciting.
Do you think that there are differences in individuals and how they respond to different bacteria? So like, would you need to screen their genomic information before you would deliver any, like how personalized do you think this sort of therapies will be? Because I wonder, maybe in a cohort of patients, 10 or 15, you might find positive results, but in a different demographic of patients, it might not work the same way, or it might be worse. So are you thinking about those things?
Yeah, absolutely. It's a really good question. I mean, I think the short answer is we already know this patient population, but I should just say all the population is quite diverse. Folks talk about, again, this kind of thesis of dysbiosis, where you have a healthy microbiome, and then in a disease population, you have a disrupted microbiome, and that's dysbiosis. But what is a healthy microbiome? I don't think we really know, because it's can vary based on region and ethnicity and nutritional intake and age and on and on and on. So I think that's a really challenging place to start. So we don't try to go after that at all. What we have seen from our phase 1 is that, this is a very diverse population, but we actually have to take things on from a very different perspective where, we're not trying to reshape the whole ecosystem of the gut. We're just trying to squeeze in these therapeutic strains. And what we've seen is through this kind of dosing pressure, so we have chronic dosing, that we're able to establish the right ecological niche and then also deliver the correct metabolic, function. And so the question around, how do we deal with kind of patient heterogeneity? Like I said, we see it, but I don't think it's going to be as large of an effect as I think we would predict. Now, with that said, things don't work for everybody. And my guess is there's complications beyond what we actually frankly understand. But I do think that that's one of the things that favors is that we're not trying to address this challenge of that heterogeneity. The other point I'll make on this point is that, what we're doing is we're following the lead of the ketogenic diet, which again is like, for those, in your audience that don't know what that is, it's developed 100 years ago, specifically to treat seizures. Its biggest utility is in drug-resistant epilepsy, meaning patients, which is about a third of all epilepsy patients, that don't respond to anti-epileptic drugs. So here you have a diet, which is like 90% of your calories from fat, and in this most strict form, that works across this very large group of folks where drugs don't work. And I will say, there's all kinds of genetic epilepsies within that space. So it seems to work across and over these different like genetic etiologies. So I think we're hoping that will be similar to that will have a similar effect as what we see there.
Can you share a little bit about the expected delivery method for BL001 and also ALS treatment 1 for BL002? Just wondering if it's something to take a pill or
Yeah, so right now they are oral. They are capsules that you take orally, and they protect against gastric exposure. I won't say much more, but we are working on oral formulations and alternative formulations as well.
I appreciate that.
Yeah.
I always wonder, why would anyone intravenously, what you're working on would not make sense intravenously. So I just wanted to kind of.
There are some, and I'll just say in the microbiome space, it's like, to some degree, even saying that doesn't do the space justice because it's just like all over, right? And there are some, I think the preclinical programs with some really interesting research where they're taking engineered bacteria and injecting it into tumors. And it turns out there's a local microbiome that's been observed. And so that's a really fascinating area. So, but since we want that central action in the gut, it's oral. There are others that are, of course, applied to the skin. There's also, even for gut, alternate deliveries, which I will leave it at that. So there's a lot of, I think, really amazing, applications of when you begin to look at the microbiome as this system that exists on our body is really primary to the functionality of all these different systems.
I appreciate that. And thanks for sharing, your next sort of critical inflection points, as you mentioned, the 2 trials you have going on and expecting to continue to, or you said start next year in 2024. The question is, what is Bloom Science's current stage of funding?
Yeah, so we, I'll kind of go through our whole funding history. We started with a seed round, and we're able to get a really nice grant as well from the ALS Association. We raised our Series A in 2021, and that was Leaps by Bayer and ALS Investment Fund as our co-lead. And then we had Apollo Health Ventures and Social Starts / Joyance Partners join in that. And right now we are also starting the next journey of fundraising. You never really stop. Throughout this period, we've been giving folks, updates and whatnot, but we're really right in the thick of it, especially since we just reported our phase 1 data.
Yeah, that makes sense. Like having that data from your phase 1 trial is another inflection point, right?
For sure.
For your company.
For sure.
It's important to make sure you can appropriately have people understand the appropriate value of your enterprise.
Absolutely.
How much have you raised so far, just to share with us?
Yeah, so we actually raised, so our Series A was 12 million, and then we had about 1.5 for our seed, just enough to really get us kind of established. And the next raise is a healthy raise. I won't mention the specific number, but it's really to get us through the end of that phase 2 in Dravet in particular, and fairly far along in the phase 2 in ALS as well. And I'll just say that puts us in a fantastic position to have 2 pivotal studies ready to go, which I think is, I think for the relative amount of investment that is being put into Bloom to have that opportunity is actually, fairly unique, I think, actually.
Yeah, I think a lot of drugs on the market now for either ALS or epilepsy are not microbiome focused or gut-brain axis focused. So this is a sort of a unique approach that you're taking, which is always refreshing and good, I think.
On the ALS side, I'll say we have a really nice slide that where we look at the other approved underlying, the mechanisms of other approved drugs. And we're really hitting those in addition to this really key nexus of neuroinflammation. And that's all of our preclinical data really supports that. So I think, we have this very unique opportunity because the nature of these, what are called live biotherapeutic products, that's a microbiota-based drug, the nature of a live cell is that it's multifunctional. And I think this is one of the big challenges, in particular in neuroscience and CNS drug development, is that these diseases are very complex. We like to oversimplify it and say it's hyperexcitabilities, oxidative stress, whatever it may be. But in reality, it's almost all the same levers for a lot of these different diseases, but just pulled at different extents. And so, I think being able to address multiple functional pathways for a disease is really key to overcome resistance, which we see, is to overcome the heterogeneity that you talked about, but then also to be able to expand that to a larger patient population.
What kind of partnerships, if any, are you seeking now?
Mm-hmm. Eyram, do you want to take that?
So I'll jump in here, and I think there are different ways of splitting things up. So there are those partnerships that we seek to form with other biopharma companies, but then there's also a whole other set of partnerships that we look at through the lens of partnering with patient advocacy groups, which are really important in indications such as Dravet syndrome and ALS. And then we have our academic collaborations as well. So on the collaborations that we'd like to form with pharma companies, our strategy was really to give ourselves the option to either go it alone, and there's always a nice opportunity there to become fully integrated, really leading the way with this modality, especially in the neuroscience space. However, as you know, it's a long road. It can be a very expensive road as well to set up a commercial organization, and there is a lot of value to be had in partnering with companies who have already been there and set up the infrastructure. So within that, there are really three buckets that we're looking at and that I'm really focusing on. So one is a co-development with the assets that we already have. So there's an opportunity to collaborate and really help us to bring BL001 to market. But the other opportunity that's really interesting is if we look at the microbiome or live biotherapeutics as a modality, almost inherent to the modality is a really clean safety profile. And within several of these neurological indications where polytherapy is the norm, that really creates a nice opportunity for our drug to be studied alongside the current standards of care, or even some other investigational therapeutics that partners may be developing. So that's another area that we're looking at. And then I'd say the third bucket within this class is really around working together to discover novel therapeutics in our partners' areas of interest. So you'll know that many of the leading companies are always looking for that next modality or that next approach to crack some of these complex challenges. So we've been having some interesting discussions around that as well.
Very interesting. And you can always take what you're learning now with your current programs and apply that to other therapeutic areas.
Yeah,
exactly.
Very interesting. So, having right now starting to raise and look at the, economic landscape, from your perspective, how is the current economic environment actually affecting deal flow and investments for your company as well as just general early stage biotech companies?
Yeah, I mean, I think it's, as everybody knows, it's not the best environment. I do think there's a couple of things. There's the very focused issues around current, investment within biotech. I think there's more macro factors that probably actually never materialized, frankly. I think everybody was preparing, late 2022 for a recession that did not materialize. It probably drove a lot of decision making at higher levels, maybe even from LPs. And you always have to remember that, as entrepreneurs, we always go to VCs to raise money, but they're also raising money. So it's this little bit of this, this kind of food chain. So I do think those global factors affected VCs, but you see them raising funds. So I don't think it's been a huge difference. And then if you look at kind of like the trajectories, really, actually, we're still on target for very good funding. It's just that 2021 and 2020 were exceptionally good years. So I think that, yes, funding is down from those years, but actually in the trajectory, if you look back the last 20 some years, we're still growing. So those are kind of the trends. In reality, I think what's happening, even though it makes things a little bit harder, I think it's good. I think it's good for entrepreneurs. I think it's good for the VCs. I think it's good for the overall ecosystem because I do think the bar has been raised a bit. And you're seeing, well, I'll say you're beginning to see more kind of maybe returning back to normal, but these bigger bets placed on less proven things, platform development companies with huge raises that, take another 3 or 4 years before they get anywhere. I think you're seeing much less of that. I think you are seeing an emphasis on clinical stage assets, the data, which is really what we need, in my opinion, obviously, to be focused on. So, but I'm pretty bullish on where things are going, to be honest with you. I think that people are putting money back into the system. I think we've been hearing just these last few weeks, frankly, about maybe the public markets opening a bit to some IPOs. So I think, there's a lot of factors at play right now, but I think things are trending in the right direction.
That's great to hear. Yeah, we've been hearing, a lot of 2020 to 2022 was so awesome, and now it's sort of becoming a little more challenging. That being said, the floor of the quality of companies is increasing now just because they have to. So that's a good thing.
I think so. Yeah.
We'll just have to see how it all unfolds.
Right. I mean,
And
again, I'm sorry, go ahead.
I was just going to say that if you think about biotech very broadly on a fundamental level, there are so many new platform technologies or technologies that are enabling different kinds of biotech companies.
Yes.
So generally speaking, the space is growing,
Yeah.
which leads me to believe that the market will grow with it.
Yeah.
And you're seeing basically cures in some cases for some therapies, and it's really amazing.
Yeah. So go ahead, Eyram. Yeah.
Well, I just wanted to jump in and say I was on an online summit call earlier today, and I'm stealing a quote from someone whose name I don't remember, so I have to apologize for that. But a venture capitalist basically said that there are sometimes moments where the market is ahead of the science, and maybe that's 2021 or maybe other periods in the last 20 years. But then there are also moments where the science is ahead of the market, and I think this is one of those, and I think that is pulling the investors back into the space.
Yeah.
Love
Yeah,
that.
and I was just going to say that's both of those points I think are so key, because imagine you're on the other side of it, right? You're making investment decisions, and you used to be able to okay, well, I'm going to look at this. I'm going to monoclonal antibodies, small molecules, limited biology, and now you have all this range of novel modalities, and with all those novel modalities comes novel biology. I mean, I think it becomes hard. It's a real challenge to sort through. It's what makes this industry, frankly, so exciting, though. I think is just. I do think we're at the beginning of, there's always ups and downs, but a larger growth phase, over the next couple of decades, frankly.
I agree. And if you consider the new applications just with AI and synthetic biology in general, there's just an amazing number of different applications, and it's very exciting.
Absolutely.
Kind of my final question, actually, having raised already, what do you think are some red flags that founders should be cautious about when they're considering striking a deal with investors?
Yeah, it's a great question. I always go back that finding investors is a little bit like dating. The first thing is you don't need everybody to like you. You just need to find that one person or a few people, in the case of the investment, that love you. And again, you're developing a relationship with your investor. They need to be, their responsibility is slightly different than yours as a founder, but you need to be in line on what the goal is. And the absolute goal is to develop, in the case of a therapeutic company, therapy is going to make significant impacts on people's lives. 100%, full stop. Everything else comes from that. So, it's a matter of can you actually communicate with this person? You don't have to be best friends with them, but you have a professional relationship. Can you personally, as a founder and entrepreneur, take feedback and sometimes really honest, brutal criticism? Are you willing to deliver it to your investors too? Say, hey, you're not thinking about this the right way, but then you got to back it up. So I think, red flags are, you'll understand really quickly through that process. Does this person match you? And yeah, I do think that we hear a lot investors are investing in the people. In biotech, I think it's a little bit different, frankly. I think it's really the science and the people, and can they execute that vision? Different than in tech, where sometimes it's just the people and they'll figure it out because they don't even know what they're doing at the beginning. So I think that's the really key things, is just making sure that you are really aligned on what the vision of the company is, what all the different stakeholders are trying to get out of that opportunity, but that all of that feeds into the mission, which is, developing, life-changing options for patients.
Yeah, and I completely agree. I think the word alignment is really key. And I think, to use the relationship analogy as well, you want to think what both sides want that relationship to look like. So especially in the early stage space, there's this idea of smart money and let's just say not smart money. And see, do you need access to that? You know, if all of your investors or potential investors are non-traditional biotech investors, you may be setting yourself back by only accepting them. But being with a company like Bloom and seeing the quality of the board and the contributions that we get, that really underlines the value of smart money. So I think, founders just need to be clear on what they need and who they need to bring into their wider team and network.
Christopher, Eyram, that's such good advice. I really appreciate it. And I hope our audience also, takes advantage of that advice. And I just want to thank you again for taking the time out today to talk to me and share your insights and what's going on with Bloom. For those listening, please like, share, and make sure you follow Bloom if you're interested. And is there anything else you guys would like to share before we conclude here?
My PR person would like for me to make a plug to go to bloomscience.com. But you can find out what we're doing, our pipeline, people, and really appreciate the opportunity to speak with you and your audience.
Excellent. I'll make sure to include the link to Bloom Science
Sounds
as well.
good.
So thank you both so much for your time today.
Thank you.